Hi, my name is Joel Nitta. Thanks for coming to my talk, Resolving Species Names Rapidly and Accurately with taxastand. You can find the slides for my talk at my GitHub, joelnitta.github.io/botany2022 taxastand. So as biologists, species names are really important to us. That's because species names are the glue that connect datasets together, whether that be specimen records or DNA sequences or trait data. In order to join any of these together, we need to do that using species names. However, this is not such a simple thing to do. And that is because of synonyms, right? So a synonym is if a species might have multiple names according to different authors. And these can be used in different data sets. And if that is the case, then we can no longer tie those together. So we need to resolve these synonyms somehow. Now, if you are only dealing with small amounts of data, maybe that's not such a big deal. You could do it by hand. But in the age of big data, doing this by hand is simply not feasible. We need software in order to resolve taxonomy at large scales. And so there's many existing software packages that can do that. But I've found that many of these have some limitations. First of all, many tools are only available via an online interface or an API. Now that can make it convenient to use, but on the other hand, if you need to go back and try to reproduce an analysis, it could be quite difficult if that API changes or if the database sitting on the other side gets updated and you can't go back to the one that you used before. Also, many of these online services only offer a limited number of reference databases to choose from. So you may not be able to implement the taxonomy of your choice for a given project. And finally, most existing taxonomic tools do not recognize the rules of taxonomic nomenclature. And because of that, we may not be able to accurately match species names. So in order to overcome some of these limitations, I've developed a new R package called taxastand. The features of taxastand include that it can be run locally in R. It allows usage of a custom reference database. So we can set it up however you want for a given project. It supports fuzzy matching, and it also understands taxonomic rules. And I'll explain what that means in a little bit more detail in some later slides. So taxastand is available on my GitHub at github.com/joelnitta/taxastand. And here I want to give a shout out to Cam Webb, who wrote a package or a software package called Taxon Tools that my R package actually uses. And if you want to find out more information about Taxon Tools, Cam presented that at Botany 2020, and I have a link to it here on this slide. So next, I'd like to demonstrate how we can use taxastand. The first thing you need to do is install taxastand. And this is the R code to do that, and it can be installed from GitHub. And like I said earlier, taxastand actually uses Taxon Tools. So in addition to that, you either need to install Taxon Tools, or you could also use a software program called Docker. Now, I don't have time to explain how Docker works in detail, but basically, if you have Docker installed, you don't need to worry about trying to install Taxon Tools yourself. Docker will take care of that for you. Okay, once we have taxastand installed and ready to use, we can do some matching with it. So this is an example of how we can use taxastand to do name matching. And that's done with the ts match names function. So that takes a couple arguments. The first one is the query, the names that we're searching for. And then the next is the reference, the names that we're matching to. So here, I'm just doing a very simple example where we've got one name in the query and two possibilities that can match to. So Crepidominus minutus matching to either Crepidominus minutum or Hymenophyllum polyanthus. And simple true means return a simple version of the output. Docker true means that we are using Docker, so we don't have Taxon Tools installed locally. And this is what the output looked like on the bottom. So it tells us again what the input was, and then it shows that it matched it to the reference name Crepidominus minutum. And you'll notice that there's a spelling difference here, right? Between the input was minutus, but it matched to minutum. So that was done using fuzzy matching. So that was an example of fuzzy matching. Next, I'm going to show an example of these taxonomic rules. So here, the query includes authors, okay, taxonomic authors. The last time it was just the bare species names. And we run the match names function again and look at the output. We see that the query Crepidonomus mutum K. ewats matched to the reference Crepidonomus mutum Bloom K. ewats. And you notice here now that the match type is different. It wasn't a fuzzy match, it says auto-Basio. And that's following these taxonomic rules where it detected that the reference includes a basionym, Bloom in parentheses, but that the input query didn't have that. So it recognized that those are actually the same name that differ only in the presence or absence of the basionym. And again, this is done using taxon tools. And I don't think there are many other taxonomic software packages that can handle that sort of thing. Okay, so those were examples of matching names. But what we really want to do is resolve names, right? Resolve synonyms. And in order to do that, we need a reference database. So taxastand comes with an example reference database, just a very small one, so you can see how this works. And this is a database of filmy ferns. So the data is called filmy taxonomy. The names I was showing earlier also happen to be filmy ferns, group that I work with. And this is what those data look like. So this is just showing a few of the columns. We have taxon ID is a unique identifier for each row. You see here we have the scientific name, and then also the taxonomic status of those names, right? So it could either be an accepted name or a synonym. And if it's a synonym, we see that in the accepted name usage ID column, that this value is mapping to the taxon ID of the accepted name. So that's really important. That's how we can go from synonym to accepted name. So you may be wondering, well, it's great that taxastand comes with filmy fern data, but what if I don't work on filmy ferns? Well, the taxonomic data that we're using here can be any data frame as long as it goes by Darwin core standards. I don't have time to explain that in detail, but you can find out more information. I have a link here. And fortunately, many existing taxonomic databases actually use Darwin core. So sources like Catalog of Life, GBIF, or ITIS, those all use Darwin core. Another source that I'm going to show in a little bit more on some other slides is ToritoCat, and this is one that I generated myself for ferns, and it's designed to work with taxastand. So in order to use your own custom database, what I recommend is probably starting from one of these existing ones. You would obtain that CSV file, load it into R with read.csv, and depending on the state of the CSV file, you may need to do some additional data wrangling. But I've actually written another package to help with that, and that's called DWC Taxon. So this has tools to wrangle data that conform to Darwin core standards. Okay, so that was a bit of a side to explain how we can get reference taxonomic data into R to use with taxastand. But assuming we have that in place, now we can resolve names. So that's done with the TS resolve names function. And we have the query as well as the reference taxonomy. And again, we're using Docker. And this is and then let's have a look at the output. Okay, so the query was Gormicormis minutum. It was matched to Gormicormis minutus Bloom-Bosch, but that name itself was a synonym you see here, and it gets resolved as Crepidonomus minutum Bloom Kowalski. Okay, so that's how we can resolve synonyms to accepted names and taking advantage of fuzzy matching and taxonomic rules, etc. Okay, so with that, I'd like to give a slightly more realistic example using the ferns of Japan. And I have the example code here available on my GitHub. Okay, so the goal of this sort of toy example is to make a map of endangered fern species in Japan. And I do that using two data sources. One is the green list, which is a list of conservation status of all fern species in Japan. And the other one is GBIF, which includes their distribution data. The problem is that green list and GBIF do not use the same taxonomy. So how can we merge these together? I was able to merge them together by matching the names of each one to a taxonomic reference called Teridocat, which is the reference database that I mentioned earlier. Now, the reason I use Teridocat is because it includes a large number of synonyms. So that's really important for resolving names. Okay, so we match names of each to Teridocat, and then we can merge those together. And finally, I've compared these results with an earlier study, Ebihara and Nitta 2019, that was a much more careful study and actually didn't use GBIF data. So we can use that to sort of ground truth our results. So most names in the GBIF data were successfully resolved. Of 1,092 species in the GBIF data, 765 were successfully merged with names in the green list. And this table is a summary of the ways that those names were matched. Okay. Now, you may be wondering, well, what about the other 300 names? Well, as it turns out, the actual total number of non-hybrid native fern taxa in Japan is around 700. So I think that those extra 300 names, many of them might have been artifacts in GBIF, perhaps non-native species or incorrect records that shouldn't have been there. And we can look at distribution maps and compare it to this previous study to see how close we got. So the top two rows are the results of this toy example where we resolve names in the GBIF data with taxastand. And then the bottom two rows, excuse me, the bottom row is data from Ebihara and Nitta 2019. Left is distribution of all species. Right column is the distribution of just endangered species. So if you just kind of squint your eyes, you can see that these do look pretty close. You'll notice that the endangered species map, that the richness does seem to be slightly higher. We have more sort of orange grid cells than in the Ebihara and Nitta data. Now that could be due again to artifacts in the GBIF data. And also notice that this is on a log scale. So even really small numbers of richness is going to show up as an orange dot here. Okay. So in summary, I think that taxastand allows for reliable, customizable taxonomic name resolution. And the biggest feature is that you can use a custom taxonomy. So if existing databases don't cut it for your project, you can prepare one for your own. So it's highly adaptable. The disadvantage of that is that doing so is not actually straightforward. So it does take some work to prepare and maintain that reference database. So in summary, I would say that like any other kind of analysis, you should just choose a tool that works best for you. And this paper here is a nice summary of recent taxonomic tools in R. So with that, I would like to say thank you to all of the institutions and individuals that made this possible. And of course, you all for listening. Thank you very much.